Welcome to ASAP Protocol Particulars, the video interview series where we meet with authors of ASAP CRN protocols. Today, I'm talking with Daniil Berezhnoi, a staff scientist on Team Wichmann. For today's episode, we're going to be discussing your methods paper, Open Source Platform for Kinematic Analysis of Mouse Forelimb Movement, which was published in STAR Protocols last month. Before we get started, Daniil, can you briefly tell us about your ASAP project and how the protocol relates to the project?
Yeah, hi, Dana. Sure. I'll start right away. The thing is, when we're talking about the circuitry mechanisms of Parkinson's disease, first thing that usually comes to mind is like neurodegeneration of dopaminergic neurons in the midbrain, or a decline in the functioning of basal ganglia, heavily relied on the dopamine neuromodulation. And all of those mechanisms imply that the activity in the motor parts of the brain, especially motor cortex, coding for complex actions, should be affected. It is kind of a textbook truth. But strangely enough, we still don't have enough studies looking closely into the cells in primary motor cortex and showing what's happening with the activity in the large pyramidal cells, sending the signals to the spinal cord and essentially controlling the muscles. So the goal of ASAP project is to study progressive changes in motor cortical activity in the rodent and primate models of Parkinson's disease, or in particular to study activity in single identified neurons coding for dexterous motor action, like reaching for food, and identify the changes over the course of disease progression. Our group, Dr. Hong Yuan Chu's lab, we're working on the rodent part of this project.
Great. So I think this is such an important question in the field, and I really enjoyed reading your paper. So I really am hoping you can talk a little bit more about why you created this behavioral apparatus and software. You know, what need does it address in the field? You know, for people who are trying to ask these questions about the circuits underlying these, complex motor skills.
Yeah, sure. So it actually comes from the idea of the whole ASAP project, or what we're doing with mice. We're working with a genetic model of Parkinson's disease, Park mice, tracking the progression of neural activity over the course of their life. Actually, from approximately 12 to 24 weeks of age. So you can imagine it's a lengthy process. We maintain the colony of the animals and start behavioral training with them when they are really young, like 6, 8 weeks of age, before the symptoms show. We need to train and perform surgery while they're still really young. And then we're injecting them with GCAM viruses, implanting with miniature microscopes to see the identified motor cortical pyramidal cells. But before that, we needed to find the task that will fit our need. It is well known that both in humans and rodents, giant pyramidal cells directly control the movement of the fingers and dexterous action like reaching. That's why we have chosen the reaching task, which is pretty standard in the field. But as we're recording neural activity during the task, mouse is literally running with a miniature microscope on its head. So we needed to control for multiple factors here, from controlling an animal behavior, providing the reinforcement, and to the parameters of the miniscope recording. That's why we needed some kind of an automatic box with multiple sensors and a camera with a fast acquisition rate to record the movement of the forepaw and synchronize with GCAM recording. And of course, to put it all together, we had to design a full comprehensive solution for the analysis of finger movement kinematics, synchronization of behavior. So actually, we've done it to synchronize all those complicated things from calcium to behavior. That's essentially how this, should I say, like hardware software platform was born.
Great. Yeah, I know from personal experience that doing synchronizing all of those things can be incredibly difficult. So this is a really amazing tool that you guys have developed for people who are doing behavior and neurophysiology recordings simultaneously. So I feel like this paper really, when you break it down, has sort of three components. So you have a section about building the apparatus and setting the software up, running the behavioral task, and then the analysis of the data. So can we break it down into those three steps and talk about, what do you see as being the most critical steps for each of these sort of sections of the protocol? And, are there any parts that you think are particularly tricky that you want to sort of give any tips to people who might want to use this apparatus?
Yes, thanks for the question. Like there's a lot to to discuss.
Yeah, I'm
It's
sure.
like three articles in one. Uh,
Well, if you want to start with if you want to just pick one where you think that the most people might encounter the most um, most difficulty in getting this this method up and running in their lab, what would you want to talk about?
yeah, sure. Um, I think there is a lot of engineering involved in this project,
Yes.
which is hard for us biologists, and it might be tricky for some users. Um, we actually developed a whole website, like just the docs, to comment on these issues. And as we write on the website, you need some experience. It might sound scary, but it's not. You need some, at least some experience with 3D printing, Arduino programming, and Python to keep this project up and running. Um, so I would say the whole assembly of the hardware and extensive testing before really starting your experiment with live animals is crucial here. Um, of course, animal training itself is kind of tricky part, and you will have to run the pilot experiment to adjust some things in like the construction of the box or how you handle animals first to see if all suggested parameters work for your animals and your particular experimental plan. Um, so yeah, I mean, I can just say, just follow the tutorial and it will work, but I know that's probably not how it works. So feel free to comment on GitHub. We'll try to answer questions. And of course, even for our own needs, we're changing this protocol from time to time and like updating the whole setup.
Can you talk about that a little bit more? How do you make a determination? How are you going about optimizing the protocol in your own lab?
Yeah, sure. Um, so for the first part, the 3D printing of the box, it is still pretty much the same. Yeah, so plastic box with four corners. What can be complicated about that? Um, though we have changed the layout of the feeder twice, if I remember correctly, just adapting to different age animals.
Mm.
And you can imagine that the success rate of animal learning depends on those little parameters like the depth of food vials in the feeder or like how close is the food pellet to the slit through which animal is reaching. So we've worked with this for quite a while. Um, what was changed the most is the protocol, I guess, itself. the protocol in engineering sense, because in our box, when you run a behavioral task, you upload it on the Arduino controller, and then it does not everything, but most things. Um, so every time you want to change something in the protocol, like for example, add a conditional stimulus, or the speed of or adjust the speed of the feeder, or add something else, you need to make these changes in the Arduino code. We've changed it multiple times. So by now, and I hope I should check on this, but of course we understand that animal training is usually very much project specific. So I think we tried to implement a few different protocols on our GitHub page. So we provided multiple codes if you want to like change the protocol. And again, every protocol with our setup is essentially a different program that you will need to load on the device. So for the convenience of the end user, we um, provide not only the protocols used during our like development uh, process and used in our training sessions described in the article, but a general tape template which you can modify to your needs. Um, and of course, I shouldn't leave animal training behind because engineering sounds complicated and fancy. But what is really tricky is to make your animals reach. It sounds easy, and a lot of students get discouraged because it's such an easy thing. It's not like patch clamping or like electrophysiology, but for some reason this thing doesn't work, and it's usually really important one because it's the foundation of the project. So I can tell you the story about that. Uh, Before we decided we'll go with the self-paced operant procedure when animal initiates each trial, we were planning to use conditional stimulus like a buzzing sound, signaling the presence of food. But it adds a few additional steps and a lot of time to training. And with our animals, we need to accomplish training like before they turn like 8 weeks before, because we still need to perform a surgery and all this kind of stuff. Um, so we decided to go with a simpler one, which is described in the article, but it's even reflected in the design of the box itself. You can even see the uh, buzzer in some of the pictures in the article, but we don't describe it in the final paper because it just turned out too complicated for them to not only to learn to reach, but also to like hold themselves and wait for the buzzer. Um, so of course there are a lot of adjustments and behind the scenes in this protocol.
Yeah, getting mice to do what you want them to do is always
Yeah.
a challenge. I'd take patching a cell any day. Um, so a big part of what I think is really great about this is that you've provided an analysis pipeline as well. So you've sort of kind of created this protocol that's start to finish for people wanting to do this. So can you talk a little bit about the analysis pipeline? Any like things that you really want to highlight about that particular part of the protocol, or that you think people really need to pay attention to when they're trying to analyze this, complicated synchronized data?
Yeah, sure. Um, First off, I guess I need to give some credits because I can't say this is a first article describing the analysis of reaching kinematics. Um, So our main idea was to just put a lot of complicated things all together. Um, and the analysis part is heavily relying on the state-of-the-art solution in the field from the Matthews lab, DeepLab Cat. Um, so I can't say like all the credit for the analysis pipeline goes to us. What we did, we just tried to put it together and add what was missing, like the analysis of kinematics itself. Mm, so I wouldn't discuss the problems that you can experience with Deep Lab Cut because they're covered on their GitHub and they're fantastic papers. What essentially we're doing, we're taking the data, CSV tables, from the Deep Lab Cut, and then using another package, which we did not develop, AnyPose, we do the triangulation, like putting two CSV tables from two not fields of view, from two views of the paw, and restoring the 3D kinematics of the fingers. And then it's pretty easy from there. uh, but so what I want to say is all our code is in multiple Jupyter notebooks, and extensive instructions are provided in each notebook. So it's supposed to lead the end users through the steps, including actually those like first steps. It opens the deep lab cut. It takes some of the code from AnyPose, and for the kinematic analysis, it just visualizes the trajectory, lets you choose the points of interest, and calculates all those parameters we're usually looking for, um, like speed, accuracy, and this kind of stuff. But for us, it saved a lot of time.
Yeah, I think one of the things that I think is really, I want to commend you on for this paper is that, I think it really exemplifies our open science ethos here at ASAP, that, you really shared every step and from start to finish in this process, and including, the Jupyter notebooks in every single step in your analysis. And I think, that's really important for other people being able to um, use this technique in their own experiments.
Yeah, sure, I agree.
So at this point, I'm just going to give you the opportunity to talk about, anything that we haven't already talked about that you think is important or that you want to highlight about your paper.
Um, I hope I hope we've done a good job, like putting it together. Um, I mean, as a full solution, because essentially that what was lacking in the field before. I haven't found an article like discussing all those things from start to finish. And when you're starting a complicated project, that's essentially what you want. Like, give me a cookbook, and I will try to follow it, and then change the recipe to my particular experiment. Um, and I don't want to say that everything is covered in the article, because obviously there are some like small bits and pieces that I try to share in this interview that are left behind the scenes. So as any protocol, it's still a work in progress. And I would really appreciate feedback from the open science community. Like, contact us on GitHub, visit our website, and ask questions, because there might be some things that are really easy to fix, but we have never encountered that. So we're really open to communication. So as you mentioned, like it is all open science. So that's community contribution, and the protocol will become only better. So, like, let me put it this way: if you face some strange problem, that's not your problem. That's probably ours, or something that we haven't mentioned. But because it is all open science, you can make it better, and or you can like contact us, and we'll try to make it better. So this kind of attitude, and let's continue to make good science together. I guess that would be my main takeaway point.
Great. I love it. Thank you so much for taking the time to talk with us today about your methods paper.
Yeah, sure. Thank you. And thank you for having us.
Of course.